I wish to thank the organizers. Yes. So you have had a few chances to learn the basics here. We start with some alphabet, which very frequently is the biology case. But in biology, we sometimes need to go beyond that to look at DNA or amino acids, and that has been a somewhat popular topic in recent time. And for today, I assume that the number of alleles at each locus is the same. It just makes stuff a little bit easier. So a fitness landscape is simply a function from the strings that could be genotypes then to the real numbers. And standard is that we also assume that an evolutionary trajectory requires that each mutation increases fitness. That's the standard idea of a fitness landscape. Now, sometimes, even though we have a theoretically large fitness landscape, things turn out to be very simple. So many times I have looked at antimicrobial drug resistance, antibiotic resistance, or similarly. And then we measure fitness in some way. We have some fitness proxy. The degree of resistance is reasonable if you think about antibiotic resistance, because it's so important for them to handle the drug that takes care of almost all fitness. And many times we can have a total black box perspective. Here's a genotype, there's a number, and I need not know anything about how it actually works. Here we have, even though fitness landscapes is in theory large, they sometimes act as if they were small. So here we have an oversimplified example. When we use an HIV drug, then a resistant mutation, here M184V, may appear. And then when we stop using the drug, then the genotype reverts back to the wild type. So this, yeah.
What do you mean by the fitness landscape being small? Do you mean the amplitude of...
Oh, no. I just mean small L. Oh, yes, the dimension, yes.
Additive support space.
Yes, that's all. So here, in this case, we could just think of, we use the drug, and then we go from 0 to 1, and then we go back from 1 to 0. So L equals 1, smallest possible dimension. And again, this is oversimplified. Now, what happens just in general if L increases? What is the impact on accessibility in the landscape, reversibility? If you go from environment A to environment B and back again, is there any hope that we find the old wild type again? How does that depend on L? Another question is, if you look at the effects of recombination, if recombination can be advantageous or detrimental, does L have any impact on that? And these are, of course, questions. Everyone has opinions about it. When it comes to accessibility in biology, it's very popular that people believe that if L is high, then accessibility is extremely good. And that comes from many times almost a caricature that people say that, okay, we draw, the worst thing we can do is to assign fitness random. So we draw it from a uniform distribution, and then obviously bounded between 0 and some value. And now if I let L be large, obviously accessibility is fantastic. Every genotype is basically one mutation away from perfection. And that is, of course, mechanistically not realistic. And you can see variants of this type of argument that we say, okay, we assign fitness values randomly to all genotypes that are not degenerate in some sense. But
I'm sorry, I don't understand you.
Okay.
If you take this fitness completely randomly on your
yes.
cube?
Yes. people do that sometimes in arguments. They check empirically what genotypes are reasonably functional and which ones are defect. And then they try to see how can we move in this landscape, and then they assign fitness values randomly, this type of argument. But let's not talk too much about that. And then what I feel very convinced about when it comes to effects of recombination, that chances that recombination is advantageous is much higher if we look at larger values of L, if we increase the dimension. So those are my opinion. And here, this I think I can go over rather quickly, since it's similar to Joachim's talk, that with the if we are in the biallelic case, then we can think of L equals 2. Then we think of the genotypes as just being the vertices in a square. For L equals 3, we get the 3 cube, and in general, an L cube. Things get a little bit different if we have the K letter alphabet. Here, K equals 3 and L equals 2, for example. Then we get 9 genotypes. And this we can think of as the product polytope of 2 triangles. And in general, if you want to use the polytope perspective, we get the product polytopes that can be described as an L-fold product of K-1 simplices. And let us now go look a little bit closer at what the fitness graphs can tell us at this point. I hope everyone is comfortable with the fitness graphs. Each arrow points toward the genotype of higher fitness. In this case, 111 is the peak. And it's very simple because we can describe this as the more mutations, the better, right? And this is compatible with additive fitness also. In contrast, here we have the opposite extreme. We have 4 peaks. And as you can see, we are always one step away from a peak. Another feature of this is sort of the efficiency, that every known peak is a source. So that would be the most extreme graph in terms of many peaks. Any questions at this point, or are fitness graphs reasonably clear? Okay, and then just to go over this dimensionality course, Dimensionality 101, that what's interesting here is that if you just look at the standard observation, that the maximal peak density is 50% if you look at this small cube, the 3 cube, and the maximal peak density is still 50% if we have L equals 1000 or any L. The maximal peak density does not change with L, right? But on the other hand, if you look at the random landscape, then the peak density changes dramatically. So there, my suspicion is that random fitness landscapes have had a little bit too much impact in how people think about real fitness landscape, because we obviously have lots of structure. So let us take a little look at fitness graphs with maximal peak density.
I mean, a comment on that. So if you think about the fitness landscapes without any prior evolution, versus if you think about fitness landscapes conditional on a whole lot of prior evolution, those are different, right? So it could have been that if you think you start with your favorite random model, you evolve for a lot, and then ask what are the statistics of what you've got after you evolve.
I see. Yes.
So you're looking at local behavior around some genome.
Sure, but I'm talking about applying that perspective on sort of empirical data from a natural population, and then you've had lots and lots of evolution, right? That's what I'm talking about.
Is that proper? Can you go back one slide?
Sure.
Is this so much of a problem in that the number of states is exponential in L, right? So even 1/1000, that is still a lot of peaks.
The number of states in what sense? I mean, in the biallelic X, we have 0 or 1 at every genotype, or I don't know.
Sequences is 2 to the L.
There
Yes.
still are many peaks.
The state space,
And if the density is low, there are still lots of peaks. I mean, that's what
2
it
to
says.
the L, K to the L.
So let's look at maximal peak density, what it looks like. That will mean that every non-peak is a source. And this, of course, the case we already had. And we just keep, and that should tell you why it is always 50%, right? We can just alternate peak and non-peak. So let us go over to instead the 3-letter alphabet. Then it's a little bit less clear how to draw the figure here. This can 0, 0, and 0, 2 are obviously equally close as 0, 0, and 0, 1. So we cannot draw a perfect figure in this case, but I think this rather well illustrates what it looks like. And this would be maximal peak density, right? We can make a third of the nodes being peaks, but obviously the distance between 2 nodes have to be 2 for both of them to be peaks. So this is maximal peak density. And here are some, if we go to K equals 3 and L equals 3 instead, then my student, Sophia Sultan, made these figures. Actually, I don't know how well you can see it, but that's what it looks like. Every genotype has 6 neighbors. And maximal peak density would look like this. I think this gives some intuition for what happens when L increases in some sense, that you can sort of keep packing peaks in some sense. So that is as much as I have about the dimensionality considerations. I will come back to that a little bit when we talk about recombination. Now, in addition to making pictures and try to get some intuition for what's going on, we also want to find exact information from fitness graphs and rank orders, or from partial orders in general. And why would I be interested in that? Well, fitness cannot always be measured. Sometimes it's easier with relative comparisons, or such comparisons are more reliable. You can think of competition experiments, for example. Another reason is that, so when in a lab we try to measure, we want to find out what the fitness is, we can almost never measure fitness exactly, which would be the quantity that the population talks about, right? We have to use some sort of fitness proxy, and there are many ways to do that. And for every time we measure, we are pretty likely to get different interactions. But then the question comes, which one is relevant? Which one do I care about? And I initially found that really annoying, especially coming from pure mathematics, that I didn't really know what everything meant exactly. And should we switch to log scale? How should we manipulate those numbers? And also, I was troubled that the numbers always came out differently, if they were sensitive for temperature or for other factors. But reasonably frequently, rank order seemed to be somewhat reliable. And then I thought, okay, but what conclusions can I draw from rank order? So that was a little bit the starting point that I got interested in fitness graphs and rank orders.
Can I ask a question?
Yeah.
How often do you find that these things are non-transitive? Like, so A is better than B, like in practice, and do you have a theory of that? Because
No, I don't. It just happened. In some settings I have observed, it was reasonably well ranked orders. That's all I can say about that, especially for antibiotic resistance.
But you mean intransitive, you change the condition, what you're measuring?
no, A is better than B, B is better than C, but then C is better than A.
Oh, I see.
But if you're measuring one quantity, what do you mean?
I mean,
Yeah,
yeah,
that's-
growth. Oh, I mean, but you
Okay, no,
compete
that-
them
yeah.
in competition.
Yeah.
Competition.
Sure.
I've not been involved in many competition experiments. I'm mostly talking about real simple measurements. Okay, so it's rather elementary that we can see that we can draw some conclusions from a rank order. If we have the form here that measures epistasis in a biallelic 2-locus case, then obviously if we have the rank order that the double mutant is more fit than the wild type, and those are more fit than the single mutants, there has to be epistasis, right? Because the two, the positive coefficients are for the wild type and for the double mutant, then obviously there has to be epistasis. Any objection to that? Okay, seem not. Three-way epistasis is measured by the form I just we have here, where you can see that we can think of this as even minus odd. All labels that are even have positive coefficients and the others have negative coefficients. Then it's also easy to see that from a rank order, for example, if all the even genotypes have the highest have higher fitness than all the odd genotypes, then obviously I would infer that there is a third order interaction from that. And now there's a very simple theorem here that this is what you can do. You just write your rank order if you have a complete rank order. And now you make a word of that, E for even and O for odd. Look at your word. If that's a Dyke word, then it implies higher order epistasis. We can also, of course, change the rules of E's and outs. And if someone has forgotten what the Dyke word is, it just means that you read from left to right, and count the number of E's. You should always have at least as many E's as O's. And then we have a considerably harder theorem. Well, I hear very few objections today. We have a considerably harder theorem, and that is to prove. And that theorem, or it wasn't a theorem, it was a question. So this was phrased as an open problem on a conference to give sufficient. To give a characterization of fitness graphs that imply higher order interaction. And if you think again, so if we have a fitness graph, we don't necessarily have a complete rank order, right? The only thing we know is how mutational neighbors compare. So it's not the same problem. And a necessary and sufficient condition in that case is that we have to be able to find a partition of the genotypes, even and odd in each one, so that we can see that the even is higher than the odd. In other words, there's some path from the even to the odd. So in this case here, the pairs would be, we can pair them so that the wild type is higher than. Oh, have I reversed the thing? The wild type. Okay, here the odds are obviously about the evens. Yes, the odds are about the evens. So and that is also that's sufficient and necessary. How can I prove that?
Do you mean a path from any E to any O, or the EO within a pair? When you said it has to be a path from E to O.
Within a pair, yeah, within each other. Okay, so can someone say how does one prove that? What theorem does it fall back on?
If you did with the wrong one.
Any guess?
Will you write down the definition of U? You can try this minus that. Is that correct?
So it actually follows from Hall's marriage theorem. Yes, there's a little bit of work. So it's pretty clear that it's to have such a partition is sufficient is pretty clear, but the hard part is to show that it's necessary. Okay, and here we have something from reality. This graph is from, let's see. So that's from Hartle's lab. It is from malaria drug resistance. And in this case, we can see that we have at least subsystem with third order interactions. If you look at, if you analyze the arrows, so to say. In this case, the 1111 has highest fitness, 0000 has lowest fitness, and it's, what do I mean by red arrows? Can someone guess what I mean by red arrows?
Ones that aren't black.
I have to be able to say what arrows are marked red.
They run away from the fittest.
They run away from the fittest, yeah, you can say, but how do red arrows differ from black arrows in this figure?
They go down.
Downwards! Brilliant, okay. Brilliant, so toward the wild type, okay. Oh yes, we use this result also for a little bit of classification of fitness graphs, and there you can see that whether or not there are higher order action is truly important for behavior. So here we looked at fourth order interaction. We look at all four locus fitness graph, and then you can see that if you look at graphs that have one peak only, then 9% of them have fourth order epistasis, and at the other extreme, if you look at graphs with 8 peaks, then all of them, well, it's just one single graph has higher order epistasis. So in other words, whether or not there is higher order epistasis will impact evolutionary trajectories. So even if you don't care so much about that in itself, it really has a role in evolution. Okay, and since we proved, my group and and another group also, since we managed to prove the theorem I stated earlier, I had hoped to have the same result here. So namely that someone comes back with a proof. So the challenge for you would be to find an upper bound for the number of peaks we can have in the absence of higher order epistasis. So we have at most, we have only pairwise interaction here. And then if you look a little bit about, if one looks a little bit about it, I have a conjecture that may very well be wrong. And that would be, but I have found graphs with that number of peaks. So it's L choose and the floor of L over 2. That would be my challenge. Now, in addition to, if you want to analyze all aspects of evolution, we obviously need more than graphs. I'm currently looking at recombination, and then I need fitness graphs, but I also need some idea of curvature because the effect of recombination is very sensitive for curvature. But I should first show fitness graphs that I used for analyzing recombination. And here, I have some line segments, so that's a little bit unclear exactly what happens there. But here you can easily, through just mutation, come up, move all the way to 1100 and 0011. But all the triple mutants have low fitness. So you can see that in such a setting, it could be tremendously advantageous with recombination because we can fly, right? Can't walk to 1111, but you could combine the two. And a huge part of historic literature on recombination works with the two-locus system. So a lot of interesting stuff happens if you look at higher L.
Can I ask for recombination? Are there breakpoints, or are you allowing arbitrary in the math literature?
You can think of it as totally independent. So the child is derived by you sort of pick randomly from
Like huge number of chromosomes.
And there's no correlation structure in how you recombine. It's just I can take.
It depends. I mean, right now my main point was just that a lot happens already. We certainly consider the situation where it's totally independent as well as linked or whatever you want. Now, for analyzing recombination, graphs are completely insufficient because graphs don't tell us anything about curvature. And now we again go back to this, we can think of a form that tells us if that describes epistasis, and if U here is greater than 0, we usually call that positive epistasis. If it's negative, it's negative epistasis. Oh, I have made a mistake here. Okay, one of the 00 should be 11. Now, what can we do instead if L equals 3? Oh, I should first say that if we have an all arrows up graph here, then recombination is, the simple description is that recombination speeds up adaptation if U is less than 0 and slows down if U greater than 0. And we can analyze this by looking at the triangulations of the square that we talked about before. And how does it work? Well, the height here, the height coordinate above the genotypes are measures of fitness. And you can see in the left there, the 1 is relatively high, and in the right 1 is relatively low. And if we think about the upper sort of roof of this building, the upper envelope of this polytope, and we project them down onto the square, we get a triangulation of the squares. And those triangulations can be quite informative about recombination. Now, this can be done in higher dimension as well. So if we have cubes, we have to triangulate the cubes. So we have to divide them into tetrahedra, again, from the values of the fitness landscape. So this works for any dimension. Yeah. Okay. This works for any dimension, and it works also for, any KNL. Now, when we go up to the 3 cube, we can no longer do only U for analyzing, for a complete analysis of the curvature, so to say. We want to look at all minimal dependence relations. And you can see A here, the first would just be a side of the cube, right? that has to be part of the picture as well. And but in addition to those sides, we also have the long diagonals in the cube, and we also have bipyramids. Of course, the circuits here, as we call them, they are all 0 if fitness is additive, and the signs of those circuits, that's what determine the triangulation. Now, nowadays at least, I don't look at all those circuits. I rather look at selected circuits for being able to analyze recombination. So that would be my current project, to try to understand recombination better. So far, what I realized, have seen that in several different ways, you have many phenomena when we increase L that make recombination more advantageous. There are some classical results that applies to all L, but they assume weak selection. And since I have worked a lot with antimicrobial drug resistance, I'm used to more, well, I cannot assume weak selection, so it gets a little bit different. Now, the first paper that works with this polytope perspective is from Bierwinkel, Tochter, and Sturmfeld, 2007, I think. Fitness graphs, you already saw, I think one of the, maybe the first fitness graph came out before I did mathematical biology. I was still doing commutative algebra in those days, by De Visser and Krug. And then I think that it really is, correct me if I'm wrong, but it really caught on in the sense that lots and lots of people wanted to present their empirical data using those graphs. And that is, eventually it reached me, and I thought that I should use those graphs to analyze, to study them as objects in themselves and analyze various theoretical problems. Sometimes we want to relate what we can see, what we can measure in a lab with something more global, and then therefore it's interesting to work with fitness graphs to sort of relate. Well, one famous result is that if we have several peaks, then we need to have also reciprocal sign epistasis, which is looks like the graph I draw first. 2 peaks, even though it's just 2 loci and 2 alleles. And then there's also, I think, the first papers that look at the interplay between fitness graphs and polytopes with Joachim and Malvika Srivastava. And it happens to be the case that you can't, maybe better name would be interplay between fitness graphs and triangulation. The fitness graph will restrict what a possible triangulation is and vice versa. So that's a rather fun math problem. And before I end, I would like to go back to the challenge that I would love to see a proof of an upper bound for how many peaks we can have unless we have higher order interaction, and that's probably a good place to end.
So there's at least no counterexample to this. So in that sense.
So I just wanted to make a comment from sort of theoretical side on thinking about, you know, natural or unnatural landscapes. And so there are nice classes of random landscapes with fitnesses chosen in Gaussian way, but with correlations in them, where one can hide large L, and where you can ask questions like, you evolve for a while, you go up here for a while, and then you look at all the statistics of the local neighborhood, multiple things there and epistasis and things there. And I think those would be really interesting ones to try to to study study more, because I think they're a better caricature of the kind of things that we is actually interested in, and they one hope that there's some advantages of it being high dimensional, and so some features may be somewhat more universal. Probably not features associated with very interesting parts of the properties of the cell, but just with basic things like the growth rates or heat tolerance, various things like that.
Is there a thought about this case where actually you frequently have with the HIV drug, where you have like one mutation seems incredibly small, right? You have a huge L, and it boils down to that usually you have one and the same mutation that goes to fixation, and then the drug is gone, it goes back again.
Yeah.
I mean, that's how on earth can you do? Can you understand that?
Well, that's time-dependent environments with very strong selection. But for example, antibiotic resistance, right? So Dan Anderson did these lovely experiments with low-dose antibiotics, and he found that you evolve cells for a long time, and they go off and get mutations, beneficial mutations in all kinds of different genes that are not normally thought to do with antibiotic resistance. And then you take the antibiotic away, and there's essentially a measurably small loss of fitness. But you have produced also some that can survive with much higher antibiotic resistance than they've ever seen. And so I think that's actually much more like the natural kind of selection that goes on more of the time, is things which are pushing in lots and lots of directions as far as the proteins are concerned all of the time. And then there is probably effects of adding a little bit of this, a little bit of this, and so on. And so a sort of random caricature of it is probably a reasonable starting point. And then, of course, the deviation from that may be some of the most interesting biology, but I think as a starting point, it is there. But one really does need to do the conditioning on a whole lot of past evolution before looking at the local statistics of the landscape.
Also, if I may follow up, because enumerating all these parts, I mean, so you presented these very nice examples for K equals 2 and L equals 2 and 3 and 4, so I look at these plots for a while and I eventually can follow. But what do you do if you have an amino acid chain where you K is of the order 20 and L can be several thousand? You cannot do these enumerations. So, well, I don't understand how you extrapolate what you learn from these enumerations in very small systems to understanding a large system without big statistics.
I mean, sure. Well, I mean, we do know some. I mean, we still have some theoretical results, right? I mean that you can't have, for example, well, I go back to the same result, but we can't have multiple peak in the large landscape unless we have certain feature that we can observe in the small landscape. So to relate to relate global properties, if you want, to local properties, that we can do, and that doesn't matter if K is 20, we can.
But I think the relevance, once you start getting large L, the relevance of number of peaks goes down. You can make models which are the same. You change one parameter, you change the number of peaks.
I think so. I mean,
but the evolution
not the number of peaks, but the
fraction.
Peak density.
No. You can explicitly write down models, which again is sort of in a natural class, where one of them has much higher density of peaks than the other, but the long-term evolutionary trajectories are similar.
Well, it's still very interesting to relate global properties with local.
It is still local property, but I think it's related to the high-dimensional continuous landscapes that various people have talked about, where in the high-dimensional landscapes, a lot of your dynamics is coming from being near saddles and so on. Right. And so that's being near peaks which are neither maxima nor minima, and all of your dynamics goes on those. Eventually, you get stuck on a peak. Yeah. You get to a peak, but but I think it's very different ensembles and that kind of natural ensembles in a loose sense, and obviously you should cook up other ones. But I think for a lot of these ones, the random one is a sort of random ones are sort of a null model, but one doesn't need to do at this point the random ones conditioned on prior evolution. I think that.
Can I ask? So you showed this beautiful table with the enumeration of all the fitness graphs that enforce a certain type of stasis, right? But that was for L equal to 3 or L equal to
Actually, L equals 4, but we did it for L equals 3.
But so for that case, you have the answer to this, right? Because you just need to sort of forbid all the graphs that enforce, you know, the stasis and I, and then you have an ensemble which has. So for that case, this is true. But as you said, I mean, you cannot really do the enumeration. Okay, any more questions? Not, then let's thank Kristina.